Hello, this is Mr. McLean, and today I want to take you through some of the early parts of the photosynthesis chapter. We're going to focus in on some of the different structures, and we want to show you how the light reactions are going to be critical for building the components that we will need later in the Calvin cycle. And I'm sure it'll be a later video where I'll put together the details of the Calvin cycle and ultimately how we're able to produce our carbohydrates. So I think probably one of the best ways to start with this is actually to jump right ahead and take a look at the reaction that photosynthesis is. And I know that we just finished up with cellular respiration in a previous chapter, and we saw that that was a reaction where you were going to be starting off with a glucose molecule, and you're going to have the situation where you're going to be ultimately breaking that scenario down and getting products coming off of it. So if we go through here and we take a look at the concept of photosynthesis, we're going to see that we definitely have an investment of both water and carbon dioxide, and the outcome is that we're going to be able to produce a glucose molecule as well as some oxygen. And if you take a look at this particular reaction, the way it's settled, a lot of times you'll see it written where water is only listed on this side of the reaction, but you're going to notice that this value is listed as 12 on the left side of the reaction and 6 on the right side of the reaction. So a lot of times with a simplified version, you'll see 6, 6, you'll see the investment of light, you'll see 1 glucose and 6 oxygens, and you won't see the water appearing on there. All right, so just so that we understand where our structures are taking place at, or where these processes are taking place at, I think one of the first things that we should take a look at is how our cells are able to actually acquire carbon dioxide. And if we look at it, what we're going to notice is that the underside of the leaf, there's going to be this opening, which is what's referred to as our stomata. And the stomata has 2 types of cells that surround it known as guard cells. And the idea of the guard cells is that they're going to help regulate whether or not that stomata is open. So if we look at it, in the case where the stomata is open, it's going to allow for the CO2 to be able to go into the cell, and then eventually it's going to get into the spongy mesophyll component, and we're going to be able to use that as we drive the Calvin cycle. There's also the possibility of water loss coming out of there whenever that's opened up. So that creates some other difficulties depending upon the environment in which the plant exists. So when we start looking at alternative pathways, for example, crustaceous acid metabolism and C4 pathways, a lot of times they're trying to accommodate for the fact that under harsh, very warm, arid conditions, a lot of times you have the potential to lose too much water. And if we take a look at some of the other things that are going to be critical, so we have our guard cells, here's the stomata, here's the opportunity for the gas exchange to take place. We have vascular tissue. Okay, so if we look at the vein of the plant, we're going to see that we are going to have the xylem and the phloem, and they're going to be surrounded with a specialized cell type known as a bundle sheath cell, which we will talk a little bit about later as well. And ultimately, I kind of like this slide because it gives us an opportunity to kind of zoom in at each level to see where we're specifically seeing these reactions take place. So this image shows us obviously a leaf, and then when we slide down to the leaf and we zoom in and we do a cross-sectional view of the leaf, what we're going to notice is that we have these structures that we've just identified, such as your stomata, where you have your gas exchange taking place, you have your vascular tissue, and you're going to have your mesophyll cells. So these things right here, these are examples of mesophyll cells. So if we zoom in, this is what we would typically think of as a plant cell. And the thing that would really jump out to us is obviously the green structures that are very obvious and prevalent. Those green structures, if we zoom in, are chloroplasts. And this is the level where we do a lot of our discussion. So when we look at the chloroplast has a lot of similar characteristics to the mitochondria that we studied during respiration. It has its own DNA. It has ribosomes. It's definitely looked at as a structure that was part of the evidence of the endosymbiotic theory, the idea that this was a standalone organism at one point. Inside the chloroplast, which in and of itself has a double membrane, what we're going to notice is that we have this environment known as the stroma. Okay, the stroma is where the Calvin cycle, so our Calvin cycle is going to take place inside the stroma. So we're going to have a lot of gas exchange that's eventually going to make it there, and we're going to have some conversion processes that will occur. The other really, really critical structure in this diagram right here is the thylakoid. And you're going to notice that the thylakoid is a structure that's somewhat oval like this, and I'll zoom in a little bit further. And as the thylakoid stacks up, so you kind of see that these are stacks of thylakoids. The stack itself is referred to as a granum. So when you hear the term granum or you read the term granum, what that's really looking at is the stack of thylakoids that are kind of working together to sort of set things up that way. All right, so if we zoom in a little bit further, that's kind of how we've gone. Now, this particular picture right here shows us a depiction of what the thylakoid looks like. And the thylakoid has a couple characteristics that we should be familiar with, one of which is the familiar phospholipid bilayer. Okay, so we see that we have a double membrane that surrounds the perimeter of the thylakoid. And obviously, the orientation of how the phospholipids are set, you're always going to have your polar heads facing outward in both directions, and your nonpolar tails are facing inward. We also have what are referred to as our photosystems. We're going to talk quite a bit about this in the light reaction section, because this is where the light is actually going to do its work. So we're going to have two photosystems. This is a photosystem which we are going to refer to as photosystem number 2. And this one right here is the second photosystem, which is actually photosystem number 1. We're going to talk a little bit about why the naming of that exists kind of in a backwards way. But basically, they discovered this process initially. And we also have some older, from an evolutionary standpoint, older organisms that only utilize photosystem number 1. So they're going to have what we refer to as cyclic electron flow, and they're not going to be able to use photosystem 2. But when we think about the role that light plays in photosynthesis, and we think about being able to ultimately produce those sugars, we have to look initially at the thylakoid, and that's a critical structure. The other thing that I want to point out to you is you're going to notice that we've created this hydrogen gradient, okay? And this hydrogen gradient is occurring inside what's referred to as the thylakoid space. So as we split things like water, we have our water molecule that's going to come in, and there's going to be enzymes that are going to help facilitate the breakdown of the water molecule. We're going to have availability of hydrogen ions. We're also going to be pumping, very actively pumping. So this right here would be an example of a proton pump. Okay, that's a proton pump. And what powers that proton pump is the downward flow from the electron transport chain. So as we examine in more detail the light reactions, we want you to make sure you understand that it's that downward flow of electrons through the electron transport chain carrier molecules. The exergonic flow of energy is going to be coupled to pump hydrogen to build this large gradient of hydrogen inside of here. And then, much like what we discovered in cellular respiration, we have a process known as Chemi Osmosis. Okay? And when you think about chemiosmosis, what's happening here is you have this accumulation of hydrogen ions that are at a very high level. We have evidence of this because we can check the pH. And if you think about what happens from a pH standpoint, the lower the value, the higher the hydrogen ion concentration. So low pH gives me high hydrogen concentration, and that's basically what we're going to see. When the light reactions are powering this, we're going to have a lot of hydrogen ions, which is proven by checking the low pH. As that hydrogen diffuses down its electrochemical gradient, we have the familiar enzyme right here of ATP synthase. Okay, so ATP synthase is the same enzyme that we found embedded in the inner membrane of the mitochondria, and it has the same function, and that is to allow the hydrogen ions to move from high concentration to low and to turn that internal rotor of sorts. And when it does that, we're able to couple that downward flow of energy to the phosphorylating process of taking ADP plus ATP or plus phosphate to generate ATP. And that ATP is actually being generated on the stromal side. The other thing that we're going to notice, and this is kind of an overview of everything, is that there really are two main products that we get coming out of the thylakoid. The first one is ATP, and I think we've emphasized many times throughout the course that that is a major energy source for a lot of cellular reactions. The other thing that we are going to get is going to be the NADPH, and NADPH is going to be a reduced molecule that's going to carry with it the ability to deliver high energy electrons. Okay, so the electrons that were initially invested in the form of H2O over here on this side of the reaction are eventually going to be released in the form of this molecule of NADPH. And both of these, the ATP and the NADPH, both of them are going to be going to the Calvin cycle. Okay, so that's just kind of an overview. Just giving you a little bit of, I know that slide is really busy now, and we have a lot of things going on that. But if this is a review for you, or if this is the first time that you're looking at it, I'm going to have a couple other video clips that will go through the details of each of these things. And we're just trying to emphasize why this is so critical. And then the underlying thing that we ultimately are going to have is that we definitely have some redox reactions that are going to be taking place throughout this entire process. So I guess the thing that you want to remember as I wrap up this introduction into photosynthesis is that photosynthesis is a two-step process. There's two major stages in what would be considered a most basic form of photosynthesis. We have what's referred to as the light reactions, and that's the part that obviously relies on light, as the name would indicate. And then we have the Calvin cycle, which is also referred to as the C3 pathway. And the Calvin cycle, named after Melvin Calvin, is when we actually produce the organic molecules. So if you look at the light reactions, we're going to see that you have investment of water that's going to be split. When the water splits, we're going to have a release of oxygen. We're going to make a product of NADPH, and we're going to make a product of ATP, both of which are going to the Calvin cycle. Okay, so both of those are going to go and take it to the Calvin cycle. If you think about the Calvin cycle, remember that this is going to be in the stroma. And I will definitely take you through a review chart of how the Calvin cycle happens. But they're going to take in the investment of carbon dioxide as well as the energy that we've organized in the form of ATP and NADPH. And they're going to execute what's referred to as carbon fixation. And that means we're going to be able to take carbon dioxide from the atmosphere, and we're going to be able to eventually make it into organic molecules. And the ultimate organic molecule that we want to see coming out of this is going to be glucose. But what you're actually going to see as we go through it is a molecule of G3P, which is a sugar as well. It's just a 3-carbon sugar. So in wrapping it up, our chloroplast is the headquarters of where this is happening at. We have the light reactions. The light reactions are going to produce ATP, NADPH. The light reactions are going to then deliver their products to the Calvin cycle, where carbon dioxide is going to be ultimately produced into sugar. So hopefully that was helpful.